Hello, I'm Pratik, and today I'm going to announce a very new series. And of course, this series is not going to publish on this channel, but another on this sub channel, and it's all about ASRB net exam, ASRB or ICMR net exam. Okay, so I'm going to tell you about the basic microbiology, basic agricultural microbiology in that particular series, and we are going to learn some basic topics and then we will practice some questions around that. So, before starting that particular series, let me just tell you that this is the first episode or this is the first lecture of that series which is publishing in this channel, but after publication of this particular lecture, I am going to teach on another channel, and I will mention the description of that channel after the completion of this video. Okay, so let's just start with the very first topic of ASRB net or ICMR net, which is history of microbiology. So let's just start it. So when we talk about history of microbiology and when we just talk about microbiology, what we imagine is little, or we can say too small to be seen with naked eyes microorganisms. They are so less, like they are less than 1 millimeter in diameter. Some are visible to the naked eye, and like for example like bacterium, Epulopiscium, or filamentous algae like bread mold and mushrooms, etc. So these are some, and you can see the picture of E. coli. We can just compare the size of the, this is the E. coli and this is Epulopiscium and this is Paramecium. So you can compare the size of these microorganisms. So we have, okay, so in that case, now, what are microbes? They are extremely diverse group, like bacteria. When we study bacteria, it's called as bacteriology. When we study algae, it's called as study of algae, like phycology. But when we study fungi, it's called mycology. Similarly, virus, when we study virus, it's called virology. Protozoa, it's called protozoology. So these are some basic terms. When we study microbiology, it actually comes with two different manner. First is theoretical or pure microbiology, and the second one is practical or applied microbiology. Both have their own significance in their own field, and both have different scopes. Theoretically, or which is actually known as the pure microbiology, is actually a study of occurrence. Anything that exists or anything that occurs in nature, it comes under this category. Second point is form, that what various forms, what kind of various alternations those species or those particular organisms is having. Third, structure. We study the structure of that particular organism with reference to previous organisms which are related phylogenetically, and then classify. Our next point, that is classification. Physiology, how they function, how the cells actually signal and perform different tasks. Reproduction, of course, how they reproduce and what strategies do they prefer in harsh conditions, how they just make endospores and all. Then heredity. Heredity is the genetic variations or genetic ability which is transformed from one generation to the next. And then variation and evolution of microbes. Of course, variation and evolution has its own significance, because without them we could not even classify the organisms. When we talk about applied microbiology, we just talk about exploitation of beneficial microbes. Like what are beneficial microbes exactly? those microbes which somehow, directly or indirectly, benefit humankind. They control the activities of harmful microbes and of course the types. When we talk about medical microbiology, it's the study of role of microbes in human and animal disease. Like we only study human body and then we study the effect of those microorganisms on human body on different aspects. Agriculture microbiology, on other aspect, we just study the role of microbes in plant disease, soil fertility, spoilage of agricultural products, and etc. When we talk about industrial microbiology, it is the utilization of microbes for the production of alcoholic beverages, antibiotics, amino acids, organic acid, acetic acid and lactic acid, citric acid, etc. So it's a commercial production of these acids by industrial microbiology. Food microbiology, when we study microbes in particularly a role of microbes in food spoilage, how microbes can spoil the food and how we can prevent the food and their prevention and increase the shelf life of various food. Like you have studied about canned foods, how do we actually manage all these things, the packaged food material and all. Production of cheese, butter, etc. Now, we are going to start with the history of microbiology, and whenever we talk about history, there is a name which always mentioned, and his name is Antony von Leeuwenhoek. When we talk about microbes discovery in late 17th century, he was the scientist or he was the observer, and that is why he is known as the one of the father of microbiology. I said one of the father because there are more which are involved in those historical background. A Dutch draper hobby was lens grinding and microscope making. Made 550 crude single lens microscope with magnification of 50x to 300x at that particular time, like 17th century. He also observed some shapes out of this microbe. So like you can just see, it was the first microscope, the Antony von Leeuwenhoek microscope. And what he observed are those cells. These were some protozoans at that time. Antony von Leeuwenhoek observation was recorded in his book, and these were some pictures or diagrams which he have prepared after the observations. Leeuwenhoek actually examines the rainwater, saliva, pepper, decoration for cork leaves of plants, seminal fluid, and scraping of teeth, etc. Out of them, he extracted the material and then he identified those microorganisms under the microscope which he has developed. He portrayed several microorganisms and called them animalcule. This was the term which was given by Antony von Leeuwenhoek for those microscopic organisms. He conveyed his findings in the series of letters, the British Royal Society, London, during the mid-1670s. Leeuwenhoek actually outlined the structural detail of paramecium, amoeba, and certain filamentous fungi, algae, bacteria, etc. Scientists also consider microbes only as curiosity of nature. Like at first, it was all about curiosity. It was all about to know a new world of microorganisms. Controversies regarding spontaneous generation, like abiogenesis. What is abiogenesis? It is the theory which was given at that time that life forms actually come from non-living material and it is spontaneously occur on Earth. This was the theory which is known as abiogenesis. This theory drew scientists to explore the origin and nature of living organisms. And spontaneous generation or abiogenesis which is actually a belief of ancient times that living organisms could develop from non-living matter, which was later on discarded by many scientists, and Pasteur was the main scientist who had discarded this completely. Theory of biogenesis, like living organisms arise only from pre-existing organism. This was also the theory at that time. There were experiments to disprove the abiogenesis process. The first experiment was conducted by Redi's, and Redi's experiment in 1688, Francesco Redi, who was an Italian physician, he carried out series of experiments on decaying meat and its ability to produce maggots spontaneously. Redi placed the meat in 3 jars. First jar, it was left open. Flies can lay egg on meat which develop into maggots. The second jar was covered with netting. Flies can lay egg on the net which was covering the jar and develop into maggots on the net. And the third jar, which is sealed pack. So there were no maggots developed on meat. So this experiment actually proved that maggots were developed from fly eggs and not from the meat, which was the belief of abiogenesis at that time. So it actually disproved the theory of biogenesis. Experiment of Louis Pasteur, which was the rule breaker, or Louis Pasteur was the one who actually demolished the abiogenesis theory. Like experiment in swan neck flask, this actually disapproves the theory of abiogenesis. Boil the flask of broth, then left it open to the air, and any of the life force, life appears. Like broth in swan neck flask, when left open in air, any of the life force, there is no appearance because of this S shape. So actually microbes can't cross this shape, this S shape, and they actually trapped in this neck. So there were no microbes in the meat broth. So this was the series of experiment. This was the swan neck, and before breaking the neck, there were no existence of microbes in the meat broth. But after breaking this neck, they actually enters. So this actually disproved that life originate from non-living organisms, non-living things. So it was disproved at that time. Then in 1800, scientists believed that infectious disease was spread by an entity, MERS, and an altered chemical quality of atmosphere. There was a scientist, Agostino Bassi, who demonstrated that a silkworm disease was due to a fungal infection. The first report that a microorganism could cause a disease. So he was very important at that time in telling us that microbes can cause disease. Then Edward Jenner. Edward Jenner in 1796 had used vaccination with materials from cowpox lesions to protect people against smallpox. So it was the first vaccine which was invented by Edward Jenner at that time. Then 1860 to 1910, the Golden Age of Microbiology was started, when Robert Koch did many research works for his things. Like many research works were conducted by Louis Pasteur and Robert Koch and others. Many branches of microbiology was established at that particular time. Here's the time when Joseph Lister, a series of discovery were mostly involving identification of agents of disease, developed an awareness that infectious disease were first caused by organism and the chain of transmission could be broken. Like he was going to tell us that microbial infection can be cured by just breaking the transmission of if we can just break the transmission of these microorganisms, we can just lead to sterilization practice in hospitals. So pasteurization of milk, purification of water, control of insects, care in preparation of food consumption, etc. So construction of a bacterial filter, which was constructed by Charles Chamberlain in 1884, he made possible the discovery of viruses and their role in disease. So the viral existence was first described by Charles Chamberlain in 1884. Then we'll talk about the Dmitry Ivanovsky. He actually discovered the tobacco mosaic virus. So he was the person behind the discovery of tobacco mosaic virus. And tobacco mosaic virus was the first viral pathogen which was studied at that time. Then we'll talk about microbiology contributed significantly to the development of biochemistry, the discovery of cell-free alcoholic fermentation by Buchner in 1897, provided the key to chemical analysis of energy yielding process. Then contribution of Louis Pasteur. He was the father of industrial microbiology too. So put an end to the theory of biogenesis, first demonstrated that the air contained microbes. He discovered anaerobic life. Actually, he discovered the anaerobic life and anaerobic form of respiration. He introduced the term aerobic and anaerobic. Established the germ theory of fermentation. Chemical changes like fermentation are vital process brought about by activity of microbes like yeast and bacteria. He established the germ theory of disease on microorganisms are responsible for infectious disease. Developed various methods of sterilization, steam sterilization, hot air oven or autoclave. He coined the term vaccine. He developed vaccine for anthrax and rabies at that particular time. Then let's just focus on contribution of F.J. Cohen. F.J. Cohen, he discovered binary fission. Binary fission, which is a common method of multiplication in bacteria. So he actually discovered the type of cell division or type of multiplication or reproduction in bacteria. Discovered endospore. Certain bacteria form extremely resistant spores like Bacillus subtilis, Clostridium botulinum, etc. Now, let's just focus on the contribution of Robert Koch in 1843 to 1910. He was father of medical microbiology because he was the first to relate specific organism to a specific disease. That means he was the discoverer of pathogens. So Bacillus anthracis caused the anthrax. He also discovers the Bacillus of tuberculosis, like Mycobacterium tuberculosis. Now let's just focus on Koch postulates. He just gave his postulates so that anybody can identify or anybody can just detect the microorganism responsible for that particular disease. So Robert Koch developed a series of procedures by which a specific organism could relate to a specific disease. These steps are called as Koch postulates. The microorganism must be in every case of disease, but absent from healthy organism was the first postulate by him. So microorganism must be present in every case. If it is present in every case, that microorganism can be the cause of that particular disease. Otherwise, it could be just a random bacteria or any other microbe. The second postulate tells us that the suspected microorganism must be isolated and grown in pure culture. Third postulate is about the same disease must result when the isolated microorganism is inoculated into healthy host. The same microorganism must be isolated again from diseased host. The same microorganism must be isolated. That means whenever we observe the disease host, that particular microorganisms which we are going to identify must be present in all the cases, like the previous postulate already told us. So, by these procedures, a specific organism could be related to a specific disease. He also developed many staining methods for bacteria to identify and to observe their structure, their nature properly. He developed pure culture techniques. He used the use of solid culture medium to grow bacteria like we use today. In the last decade of 19th century, microbiologists' interests were concentrated on the role of microbes as agents of infectious disease. So this was called as the golden period for microbiology. Since many of the important pathogenic organisms have been discovered, their causal roles established. The students of Pasteur or maybe Koch and others discovered that in rapid succession a group of bacteria is capable of causing specific disease. Techniques and laboratory procedures. Okay, so here you can see the photograph of Ernst Ruska and Max Knoll with their electron microscope. This was the first electron microscope which was used in that particular time in 19th century. So during the last decade of the 19th century, microbiology became an established discipline, so it was recognized as a discipline. Although RPSC still didn't recognize microbiology, but it was established discipline in 19th century. So maybe RPSC is a bit slower. Okay. So if we say, so during the early part of 20th century, microbiology developed independently other biological disciplines. The discovery of electron microscope was in the year 1934. So development of drug or chemicals which could kill infectious agents while being on non-toxic the host. In 1929, it was Alexander Fleming who accidentally discovered the antibiotic penicillin. Penicillin from Penicillium notatum, which destroyed Staphylococcus, the first antibiotic form. So what are antibiotics actually? Antibiotics are actually secondary metabolite which produced at the time of or before the declining phase of any bacteria. These secondary metabolites are produced just to give an antagonist effect on other organisms so that the resources can be available for that particular organisms. So this is a kind of survival method to produce antibiotic. Now as we know about it, so we can use that antibiotic, that particular antibiotic, to kill some pathogens. So this is why it is beneficial, it has the beneficial role for humankind. So many antibiotics are produced from microbes. Microbiology established a closer relationship with other disciplines in 1940s. Now microbiology is the science which can actually correlate with these other sciences. The first important connection between the genetics and microbiology is the work of Beadle and Totem. It was very important work. It was held in 1941 when they succeeded in isolating biochemical mutant in Neurospora crassa. You know, Neurospora is also known as Drosophila of plant kingdom. One of the gene in one gene, one enzyme hypothesis was given by Beadle and Totem. So use of microbes to produce valuable products and then use it in industrial microbiology was the aim. So development of Rdna technology or recombinant DNA technology, the gene cloning or genetic engineering, genetically modified microbes which are actually those various mutations which was stabilized by them. So genetically modified microbes that can be used as factory cells for the synthesis of valuable pharmaceutical products like human insulin, hormones, vaccine, etc. Then we talk about space microbiology. It is a branch of exobiology investigating the possibilities of extraterrestrial life and effects of extraterrestrial environments on living organisms from the Earth. Investigates the possibility of using microbes as source of food and oxygen. So there are many examples of those kind of foods like SCP. SCP is single cell protein which is used by astronomers so that they can feed themselves in space. Modern microbiology is a well-developed discipline with established concepts and techniques. Now, let's just talk about the scope of microbiology. Many microbiologists actually focus on specific group of microorganisms so that they could be better for any observation on particular organisms. Like virologists, they study for virus, so their main focus on viruses. Bacteriologists on bacteria and phycologists on algae. When we talk about fungus or fungal related products, so we focus on mycologists. We say them mycologists. And protozoologists on protozoa. So there are different fields in microbiology, as we can say that microbial morphology, where we study the external features of those microorganisms. Microbial physiology, where we study the functionality or physiology, or complete cycles or their metabolic rate, their metabolism and etc. Microbial cytology, where we actually study the cell signaling process or the more about cellular respiration and all. Microbial ecology, it is the science when we are able to relate microbes with the environment. Then microbial genetics and molecular biology. So microbial genetics and molecular biology is the science where we connect the microbiology to the gene level. Then microbial taxonomy, where we can classify things after giving their name. And medical microbiology, which is very essential in today's scenario, as it studies different microorganisms' effect on different type of disease and their pathogenicities. When we talk about public health, we have to talk about microbiology, for food microbiology, for daily microbiology or immunology. Microbes are extremely useful as experimental materials also. Like they are simple nature, like they are single cell, easy to grow, and they grow very rapidly. They can culture in a large quantity in test tubes. Flask requires less space and maintenance. We compare with any kind of animals, because animal has a bigger size, so we have to maintain more precautions and we have to wait for their life cycle to complete. But in case of microbes, they have a very short life cycle, so we don't have to wait a lot. So they can culture in large quantity in test tubes. Flasks can require less space and maintenance than large plants and animals. They are easy to work with, material for studying complex process for metabolism. So metabolic process is similar to higher plants and animals. The only difference is there is no labor of division or there is no division of cellular compartments, because the microbes or the prokaryotic cells is actually composed of only cytoplasm inside it. There is no membrane bound structures found there. And when we talk about some protozoans, they also have some simpler organization, even having a eukaryotic cell. So microbes can be used to study synthesis of antibiotics, toxins, energy production, survival mechanisms in harsh environmental conditions. So microbial nitrogen fixation is also a part of microbial activities. Thank you. So that's enough for today, and we are going to talk about more on agricultural microbiology on our new channel. So do support us, and if you require anything else, if you require notes or anything else, you can comment on it. So let's just end this stream for now, and we'll meet tomorrow. Thank you. So bye bye.